Chapter 1: The Insect Body Plan – Architecture for Success. Insects represent the most successful group of animals on Earth, comprising over 80% of all known animal species, and occupying virtually every ecological niche from deep ocean trenches to high mountain peaks. Their extraordinary success stems from a fundamental body plan that has proven remarkably adaptable across millions of years of evolution, enabling insects to exploit resources and environments unavailable to other organisms. Understanding insect biology begins with examining their basic architecture, which provides the foundation for their incredible diversity and ecological dominance. The segmented body plan of insects reflects their arthropod heritage, while incorporating specialized features that distinguish them from other members of this diverse phylum. The insect body consists of three distinct regions: the head, thorax, and abdomen, each serving specific functions that contribute to survival and reproduction. This tagmatization, or division into specialized body regions, allows for efficient organization of sensory organs, locomotory structures, and internal systems, while maintaining the structural integrity necessary for terrestrial life. The exoskeleton represents perhaps the most defining feature of insect anatomy, providing both protection and structural support through a complex arrangement of sclerotized plates connected by flexible membranes. Composed primarily of chitin embedded in a protein matrix, the exoskeleton serves multiple functions including muscle attachment, water conservation, and defense against predators and environmental hazards. The cuticle's multi-layered structure includes an outer epicuticle that provides waterproofing through waxy secretions, a middle exocuticle that provides strength through cross-linked proteins, and an inner endocuticle that offers flexibility and growth accommodation. The constraints imposed by the exoskeleton have shaped fundamental aspects of insect biology, particularly the process of molting that enables growth and development. As insects grow, they must periodically shed their rigid exoskeleton and secrete a new, larger one, a process controlled by complex hormonal interactions. The molting cycle involves preparation phases where new cuticle is secreted beneath the old one, ecdysis when the old cuticle is shed, and hardening phases where the new exoskeleton sclerotizes and gains its final strength. This process represents a vulnerable period when insects are soft and defenseless, requiring careful timing and protective behaviors. The head capsule houses the primary sensory organs and feeding apparatus, demonstrating remarkable specialization across different insect groups. Compound eyes, composed of numerous individual ommatidia, provide wide field vision capable of detecting movement and polarized light patterns useful for navigation. Simple eyes or ocelli supplement compound eyes by detecting changes in light intensity and maintaining orientation responses. The antennae serve as chemical and mechanical senses, with elaborate branching patterns in some species maximizing surface area for detecting pheromones and other environmental cues. Insect mouthparts exhibit extraordinary diversity, reflecting the vast array of feeding strategies employed by different species. Chewing mouthparts, considered the ancestral condition, consist of mandibles, maxillae, and a labium that work together to manipulate solid food. Piercing sucking mouthparts in mosquitoes and aphids form needle-like structures, capable of penetrating plant or animal tissues to access fluids. Sponging mouthparts in flies use absorbent pads to take up liquids, while lapping mouthparts in bees and butterflies form elongated tubes for accessing nectar from deep within flowers. The thorax serves as the locomotory center, bearing 3 pairs of legs and, in most insects, 2 pairs of wings that enable both terrestrial and aerial movement. Each thoracic segment contains complex musculature that powers leg movements for walking, jumping, digging, or grasping, depending on the species' ecological requirements. The wing-bearing segments house powerful flight muscles that may comprise up to 80% of an insect's body mass in strong flyers like dragonflies and bees. This muscular investment reflects the enormous energetic demands of insect flight and its importance for dispersal, foraging, and mate location. Insect legs demonstrate remarkable morphological diversity that reflects specialized functions across different species. Cursorial legs in ground beetles are elongated and streamlined for rapid running, while fossorial legs in mole crickets are modified with enlarged femora and shovel-like tibiae for digging. Saltatorial legs in grasshoppers feature greatly enlarged hind femora containing powerful muscles for jumping, and raptorial legs in praying mantises bear rows of spines for grasping prey. Natatorial legs in aquatic insects may be flattened and fringed with hairs to function as paddles. Wings represent one of the most significant evolutionary innovations in insect biology, enabling three-dimensional movement through the environment and access to resources unavailable to terrestrial arthropods. The basic wing structure consists of a thin membrane supported by a network of veins that provide both structural support and pathways for hemolymph circulation and nerve supplies. Wing venation patterns are highly conserved within taxonomic groups and serve as important characters for identification and phylogenetic analysis. The evolution of wing folding mechanisms has allowed insects to protect their wings when not in use, while maintaining compact body forms. The abdomen contains the bulk of internal organs, including the digestive system, reproductive organs, and much of the respiratory and circulatory systems. Abdominal segments may bear specialized structures such as cerci that serve sensory functions, claspers used in mating, or ovipositors for egg placement. The flexibility of abdominal segments allows for expansion during feeding or egg development and enables complex behavioral movements during mating and egg-laying. Sexual dimorphism often manifests in abdominal structure, with females typically having larger abdomens to accommodate egg development. Internal anatomy reveals sophisticated organ systems adapted for the unique challenges of insect life. The digestive system varies greatly depending on diet, with specialized regions for food storage, digestion, and absorption. Herbivorous insects often possess enlarged crops for storing plant material and extended midgets with numerous digestive cilia to maximize enzyme production and absorption surface area. Carnivorous insects typically have shorter, more muscular digestive tracts adapted for processing protein-rich prey. The presence of symbiotic microorganisms in many herbivorous insects enables digestion of cellulose and other complex plant compounds. The respiratory system employs a unique tracheal network that delivers oxygen directly to tissues without relying on circulatory transport. This system consists of external spiracles that control air entry, main tracheal trunks that distribute air throughout the body, and progressively smaller tracheoles that terminate at individual cells. This direct oxygen delivery system enables the high metabolic rates necessary for flight, while avoiding the limitations imposed by diffusion across body surfaces. However, the tracheal system also represents a significant water loss pathway that constrains insect distribution in arid environments. The circulatory system differs fundamentally from that of vertebrates, consisting of an open system where hemolymph bathes internal organs directly rather than being confined to blood vessels. The dorsal vessel serves as the primary pumping organ, moving hemolymph from the abdomen toward the head through a series of chambers equipped with one-way valves. Accessory pulsatile organs help circulate hemolymph through appendages and wings. The hemolymph transports nutrients, hormones, and waste products, while playing important roles in immune defense and wound healing. Size constraints have profoundly influenced insect evolution, with the largest insects being limited by the efficiency of their respiratory and circulatory systems. The passive diffusion of oxygen through tracheae becomes increasingly inadequate as body size increases, explaining why modern insects are relatively small compared to their Paleozoic ancestors that lived in high oxygen atmospheres. Surface area to volume ratios also affect heat exchange, water balance, and other physiological processes that constrain insect size and shape. Despite these constraints, insects have achieved remarkable diversity within their size limitations. The nervous system demonstrates remarkable miniaturization, while maintaining the complexity necessary for sophisticated behaviors including navigation, communication, learning, and social coordination. The brain, though small, contains specialized regions for processing sensory information, integrating behavioral responses, and storing memories. Ganglia throughout the body provide local control of reflexes and basic functions, allowing for rapid responses to stimuli without central processing delays. The organization of insect nervous systems provides insights into the minimum neural architecture required for complex behaviors. Sensory capabilities often exceed those of larger animals, with insects detecting chemical, visual, mechanical, and thermal stimuli across wide ranges of sensitivity. Chemoreception enables detection of food sources, mates, and oviposition sites at considerable distances, while mechanoreception provides information about air currents, substrate vibrations, and contact with objects. Some insects detect ultraviolet or infrared radiation invisible to humans, while others sense electrical fields or magnetic orientations useful for navigation. These enhanced sensory capabilities compensate for small brain size by providing detailed environmental information. The modular organization of insect body plans has enabled evolutionary experimentation with different combinations of features, leading to the extraordinary diversity we observe today. Basic body segments can be modified, fused, or specialized for particular functions while maintaining overall structural integrity. This developmental flexibility, combined with high reproductive rates and short generation times, has allowed insects to rapidly evolve adaptations to new environments and ecological opportunities. Understanding insect body plans provides the foundation for appreciating how these remarkable animals have achieved their current dominance in terrestrial ecosystems. Chapter 2: Metamorphosis and Development. The phenomenon of metamorphosis represents one of the most remarkable transformations in the biological world, enabling insects to completely reorganize their body structure and function during development. This process allows insects to exploit different ecological niches at different life stages, reducing intraspecific competition while maximizing resource utilization throughout their life cycle. Understanding insect metamorphosis reveals the complex interplay between genetic programming, hormonal control, and environmental influences that guide development from egg to adult. Insect development encompasses several distinct patterns that reflect different evolutionary strategies for growth and maturation. Ametabolous development, found in primitive wingless insects such as springtails and silverfish, involves gradual growth without significant changes in body form or proportions. Young individuals resemble miniature adults and simply increase in size through successive molts until reaching sexual maturity. This developmental pattern represents the ancestral condition and demonstrates how growth can occur within the constraints of an external skeleton. Hemimetabolous development, characteristic of insects such as grasshoppers, dragonflies, and true bugs, involves gradual transformation through a series of increasingly adult-like stages called nymphs. Each molt brings the developing insect closer to adult form, with wing buds gradually enlarging and reproductive organs developing internally. The final molt transforms the last nymphal stage into a sexually mature adult, capable of reproduction and typically flight. This gradual metamorphosis allows continuous feeding and growth, while slowly acquiring adult capabilities. Holometabolous development represents the most complex and evolutionarily advanced form of insect metamorphosis, involving complete transformation between larval, pupal, and adult stages. This dramatic reorganization requires the breakdown of larval tissues and the construction of entirely new adult structures from groups of undifferentiated cells called imaginal discs. The larval stage specializes in growth and food storage, the pupal stage accomplishes metamorphic transformation, and the adult stage focuses on reproduction and dispersal. This division of labor between life stages has contributed significantly to insect evolutionary success. The endocrine control of metamorphosis involves complex interactions between prothoracicotropic hormone, PTTH, ecdysone and juvenile hormone that coordinate molting and development. PTTH, produced by neurosecretory cells in the brain, stimulates the prothoracic glands to produce ecdysone, the molting hormone that initiates the molting process. Juvenile hormone, secreted by the corpora allata, determines whether molts result in another juvenile stage or progression toward adult form. The presence of juvenile hormone maintains juvenile characteristics, while its absence permits metamorphic transformation. Larval development focuses primarily on growth and nutrient accumulation, with specialized morphology adapted for efficient feeding rather than reproduction or dispersal. Caterpillars possess powerful mandibles for consuming plant material, and enlarged digestive systems for processing large quantities of food. Maggots exhibit simplified body forms with reduced sensory organs and appendages, reflecting their specialized feeding environments. The larval cuticle often allows considerable expansion to accommodate rapid growth, while behaviors focus on finding food and avoiding predators. The diversity of larval forms reflects the wide range of ecological niches occupied by developing insects. Aquatic larvae such as mosquito wigglers and mayfly nymphs possess specialized respiratory structures including gills and spiracular modifications for underwater life. Wood-boring larvae develop powerful mandibles and elongated body forms for tunneling through solid substrates. Parasitic larvae may possess specialized attachment organs and simplified digestive systems adapted for their specialized lifestyles. This larval diversity enables insects to exploit resources unavailable to adults. Pupation represents a revolutionary reorganization of body structure involving the dissolution of larval tissues and construction of adult organs from previously dormant cell clusters. Imaginal discs present throughout larval development contain the genetic programming necessary to construct adult structures including wings, reproductive organs, and complex sensory systems. The pupal stage requires protection from predators and environmental hazards, while this vulnerable transformation occurs, leading to the evolution of pupation chambers, cocoons, and other protective structures. The molecular mechanisms underlying metamorphosis involve cascades of gene expression changes that coordinate the timing and pattern of tissue reorganization. Ecdysone acts as a master regulator that activates transcription factors controlling hundreds of genes involved in tissue breakdown and construction. Different tissues respond to hormonal signals with different timing and sensitivity, ensuring proper coordination of developmental events. The genetic control of metamorphosis provides insights into how complex developmental programs evolved and are maintained. Environmental influences can significantly affect the timing and success of metamorphic development, with temperature, photoperiod, and nutrition playing particularly important roles. Temperature affects the rate of development, with each species having optimal thermal ranges for successful metamorphosis. Photoperiod cues help synchronize development with seasonal conditions, ensuring that adults emerge when environmental conditions favor survival and reproduction. Nutritional quality during larval development affects adult size, reproductive capacity, and survival, demonstrating the importance of larval feeding success. Diapause represents a specialized developmental response that allows insects to survive unfavorable environmental conditions by temporarily halting development at specific life stages. Facultative diapause occurs in response to environmental cues such as shortened day length or temperature changes, while obligate diapause occurs as a regular part of the life cycle regardless of conditions. Diapause involves dramatic metabolic slowdown, enhanced stress resistance, and often behavioral changes that improve survival during adverse periods. The evolution of diapause has enabled insects to exploit seasonal environments and expand their geographic ranges. The evolution of complete metamorphosis represents one of the most significant innovations in animal development, enabling the remarkable diversity and ecological success of holometabolous insects. This developmental strategy allows specialization of different life stages for different functions, while avoiding competition between juveniles and adults. The evolution of metamorphosis required coordinated changes in developmental genetics, endocrine systems, and cellular mechanisms that facilitate tissue reorganization. Understanding how complete metamorphosis evolved provides insights into major evolutionary transitions. Growth and development patterns vary significantly among insect orders, reflecting different life history strategies and ecological adaptations. Fast-developing species may complete multiple generations per year, but produce smaller adults with shorter lifespans. Slow-developing species invest more time in growth and may produce larger adults with greater reproductive potential. Some species exhibit determinate growth where development proceeds through a fixed number of instars, while others show indeterminate growth where the number of molts varies with environmental conditions. The coordination of development with seasonal cycles requires sophisticated integration of environmental cues with internal developmental programs. Many temperate insects synchronize reproduction and development with predictable seasonal patterns of temperature, moisture, and food availability. Tropical insects may respond to wet and dry seasons or other cyclical environmental changes. The ability to predict and respond to seasonal changes has been crucial for insect success in variable environments and has led to complex life cycle adaptations. Developmental anomalies and plasticity demonstrate the robustness and flexibility of insect developmental systems. Environmental stresses during development can cause deformities or delays, but insects often show remarkable recovery ability through compensatory growth and repair mechanisms. Some species exhibit polyphenism, where different environmental conditions during development produce alternative adult forms adapted for different ecological conditions. This developmental plasticity enables single genotypes to produce multiple phenotypes suited for different environments. The regeneration capabilities of developing insects vary among species and life stages, with younger stages generally showing greater regenerative potential than older ones. Lost appendages may be regenerated during subsequent molts, though the process requires precise coordination between wound healing, cell proliferation, and developmental patterning. Understanding insect regeneration has provided insights into developmental biology and has potential applications for regenerative medicine in other organisms. Social insects have evolved complex developmental pathways that produce different castes specialized for different colony functions. The development of worker, soldier, and reproductive castes involves differential gene expression, triggered by nutritional, chemical, or social cues during larval development. Queen bees develop from larvae fed exclusively royal jelly, while worker development results from different nutritional regimes. The evolution of caste systems demonstrates how developmental plasticity can be harnessed for social evolution. Modern research techniques including genomics, proteomics, and advanced imaging are revealing new details about insect development and metamorphosis. Genetic manipulation techniques enable researchers to investigate gene function during development, while new microscopy methods allow real-time observation of developmental processes. These advances are providing unprecedented insights into how complex developmental programs evolve and function, with implications for understanding development in other organisms and potential applications in pest management and biotechnology. Chapter 3: Physiology and Biochemistry. The physiological systems of insects represent remarkable examples of biological efficiency and adaptation, enabling these animals to thrive in environments from Arctic tundra to tropical rainforests while maintaining high activity levels despite their small size. Understanding insect physiology reveals how fundamental biological processes have been modified to operate within the constraints of miniaturization, while supporting the energetic demands of flight, rapid development, and complex behaviors. The biochemical innovations underlying these physiological capabilities provide insights into the molecular basis of insect success. Respiration in insects employs a unique tracheal system that delivers oxygen directly to tissues without relying on circulatory transport, representing a fundamental departure from the respiratory strategies employed by most other animals. The tracheal network begins with external spiracles that can be opened or closed to control gas exchange and water loss. Main tracheal trunks distribute air throughout the body, branching into progressively smaller tubes that terminate in tracheoles delivering oxygen directly to individual cells. This system enables the high oxygen consumption rates necessary for flight, while maintaining fine control over gas exchange. The spiracular control system demonstrates sophisticated physiological regulation that balances oxygen supply with water conservation, a critical consideration for terrestrial animals with high surface area to volume ratios. Spiracular muscles can close these openings to reduce water loss during periods of low oxygen demand, while opening them when increased gas exchange is required. Some insects exhibit discontinuous gas exchange cycles, where spiracles remain closed for extended periods, dramatically reducing water loss while maintaining adequate oxygen supply through stored air in the tracheal system. Flight metabolism represents one of the most energetically demanding activities in the animal kingdom, requiring insects to sustain oxygen consumption rates that exceed those of any other animals. The flight muscles of insects contain extraordinarily high concentrations of mitochondria, enabling rapid ATP production through aerobic respiration. Direct flight muscles attach directly to wing bases and power wing movements through synchronized contractions, while indirect flight muscles power flight through distortions of the thoracic exoskeleton. The biochemical adaptations supporting flight include specialized metabolic pathways for rapid fuel utilization and efficient oxygen transport. The circulatory system of insects operates as an open system where hemolymph bathes internal organs directly rather than being confined to blood vessels, requiring different strategies for fluid transport and pressure regulation compared to closed circulatory systems. The dorsal vessel serves as the primary pumping organ, containing multiple chambers with one-way valves that move hemolymph from the abdomen toward the head. Accessory pumping organs help circulate hemolymph through appendages and wings, ensuring adequate nutrient delivery and waste removal throughout the body. Hemolymph composition differs significantly from vertebrate blood, lacking specialized oxygen-carrying cells and instead relying on dissolved oxygen and specialized proteins for gas transport. The hemolymph contains nutrients, hormones, immune factors, and waste products, while serving important functions in hydraulic pressure generation for molting and wing expansion. Some insects possess hemoglobin or other respiratory pigments that enhance oxygen transport, particularly in aquatic species where oxygen availability may be limited. The ionic composition of hemolymph is carefully regulated to maintain proper cellular function and nerve conduction. Temperature regulation in insects relies primarily on behavioral thermoregulation and biochemical adaptations, rather than the physiological mechanisms employed by endothermic vertebrates. Behavioral strategies include basking in sunlight to absorb heat, seeking shade to avoid overheating, and selecting microhabitats with favorable thermal conditions. Some large insects can generate heat through flight muscle activity, enabling flight at lower ambient temperatures than would otherwise be possible. Biochemical adaptations include antifreeze proteins that prevent ice crystal formation, and temperature-compensated enzyme systems that maintain function across thermal ranges. Water balance represents a critical physiological challenge for terrestrial insects due to their high surface area to volume ratios and the permeability of their exoskeletons to water vapor. The waxy epicuticle provides the primary barrier to water loss, with specialized lipid compositions that minimize permeability while maintaining cuticular flexibility. The Malpighian tubule system enables efficient water recovery from metabolic waste, producing highly concentrated urine that minimizes water loss. Some desert insects have evolved remarkable water conservation abilities, including metabolic water production and behavioral strategies for harvesting atmospheric moisture. The excretory system of insects employs Malpighian tubules and specialized portions of the hindgut to eliminate nitrogenous wastes while conserving water and essential ions. Malpighian tubules actively transport uric acid, water and salts from the hemolymph into the gut lumen, where specialized regions of the hindgut selectively reabsorb water and useful ions while concentrating waste products. The production of uric acid, as the primary nitrogenous waste product, conserves water compared to ammonia or urea excretion, while enabling efficient waste elimination in terrestrial environments. Digestive physiology varies dramatically among insects depending on their dietary specializations, with herbivorous, carnivorous, and detritivorous species employing different strategies for nutrient extraction and utilization. The insect digestive system typically includes a foregut for food storage and initial processing, a midgut where most enzyme secretion and absorption occurs, and a hindgut for water recovery and waste formation. Specialized structures such as gastric ceca increase digestive surface area, while some insects house symbiotic microorganisms that enable digestion of otherwise indigestible materials. Enzyme systems in insect digestive tracts show remarkable diversity, reflecting their varied diets and feeding strategies. Cellulases enable some insects to digest plant cell walls, while chitinases help parasitic insects penetrate host tissues. Proteases and lipases break down protein and fat components of prey or plant tissues. Some insects produce enzymes that detoxify plant secondary metabolites, enabling them to feed on chemically defended plants. The regulation of enzyme production often responds to dietary composition, demonstrating physiological flexibility in digestive capabilities. Symbiotic relationships with microorganisms enable many insects to exploit nutritionally poor or chemically defended food sources that would otherwise be unavailable. Termites house protists and bacteria that digest cellulose, while aphids rely on bacterial symbionts to synthesize essential amino acids absent from their plant sap diet. These relationships often involve complex integration between host and symbiont physiology, with specialized organs housing microbial partners and coordinated reproduction, ensuring symbiont transmission to offspring. The evolution of these partnerships has been crucial for insect exploitation of plant resources. Stress physiology in insects involves complex responses to environmental challenges including temperature extremes, desiccation, starvation, and chemical exposure. Heat shock proteins protect cellular components during thermal stress, while cryoprotectants prevent freezing damage in cold-adapted species. Metabolic depression during starvation conserves energy stores and prolongs survival, while detoxification systems eliminate harmful chemicals encountered in food or the environment. Understanding stress physiology is important for predicting insect responses to environmental change and developing pest management strategies. The endocrine system coordinates insect physiology through complex networks of hormones that regulate development, reproduction, metabolism, and behavior. Juvenile hormone affects development and reproduction, while influencing various physiological processes, including metabolism and immune function. Ecdysone not only controls molting but also affects gene expression in various tissues throughout the life cycle. Insulin-like peptides regulate growth and metabolism, while adipokinetic hormones mobilize energy stores during flight and other energetically demanding activities. Immune physiology protects insects from pathogens and parasites through both cellular and humoral responses that recognize and eliminate foreign threats. The insect immune system includes hemocytes that engulf pathogens and damaged cells, antimicrobial peptides that kill bacteria and fungi, and melanization reactions that encapsulate larger threats. The complement system and other immune cascades amplify responses to infection, while avoiding excessive inflammation that could damage host tissues. Understanding insect immunity has applications for biological control of pest species and insights into immune system evolution. Metabolic biochemistry in insects involves specialized pathways for energy production, storage and utilization that support their unique physiological demands. Trehalose serves as the primary circulating sugar, providing readily available energy while remaining stable in hemolymph. Lipid metabolism supplies fuel for long-distance flight, with specialized biochemical pathways for mobilizing and transporting fats. Protein metabolism supports growth and reproduction, while providing amino acids for specialized biochemical functions. The coordination of these metabolic pathways enables insects to rapidly shift between different physiological states. Detoxification systems enable insects to survive exposure to plant toxins, pesticides, and other harmful chemicals through specialized enzyme systems that modify or eliminate these compounds. Cytochrome P450 enzymes metabolize a wide variety of xenobiotics, often converting them to less toxic forms that can be eliminated. Glutathione transferases conjugate toxins with glutathione for elimination, while esterases break down specific classes of compounds. The evolution of enhanced detoxification capabilities has enabled insects to exploit chemically defended plants and has led to pesticide resistance in many species. Advances in molecular biology and biochemistry continue to reveal new aspects of insect physiology and their potential applications. Genomic and proteomic approaches are identifying new physiological pathways and regulatory mechanisms. Understanding insect biochemistry is leading to new strategies for pest control and beneficial applications including pharmaceutical production and biomaterial development. The study of insect physiology continues to provide insights into fundamental biological processes, while revealing the remarkable adaptations that have enabled these animals to colonize virtually every terrestrial environment. Chapter 4: Behavior and Ecology. Insect behavior encompasses an extraordinary range of activities from simple reflexes to complex social interactions, demonstrating remarkable sophistication despite the small size and relatively simple nervous systems of these animals. The behavioral repertoires of insects enable them to locate food and mates, avoid predators, navigate complex environments, and coordinate activities with other individuals. Understanding insect behavior requires examining both the proximate mechanisms that control behavioral responses and the ultimate evolutionary factors that have shaped these behaviors over millions of years. Communication systems in insects employ diverse sensory modalities, including chemical, visual, acoustic, and tactile signals that enable information transfer between individuals. Chemical communication through pheromones represents the most widespread form of insect communication, enabling species recognition, mate location, territorial marking, and coordination of social activities. Sex pheromones can attract mates from considerable distances, while alarm pheromones coordinate defensive responses in social species. Trail pheromones enable foraging ants to mark profitable food sources and recruit nestmates to exploit these resources efficiently. Visual communication employs patterns, colors, and movements that convey information about species identity, sex, reproductive status, and behavioral intentions. The wing patterns of butterflies often serve dual functions of species recognition and predator deterrence, with eye spots and warning coloration providing protection while maintaining intraspecific signaling capability. Firefly bioluminescence creates species-specific flashing patterns that enable mate recognition in complex communities where multiple species are active simultaneously. The evolution of visual signals reflects the interaction between signal efficiency and the sensory capabilities of intended receivers. Acoustic communication enables long-distance signaling and precise temporal coding of information, with insects producing sounds through diverse mechanisms including stridulation, timbal vibration, and wing beating. Cricket and grasshopper songs encode species identity, male quality, and aggressive intentions through variations in frequency, amplitude, and temporal patterns. The acoustic environment influences signal design, with species in noisy habitats often using higher frequencies or different temporal patterns to avoid interference. Some insects use substrate-borne vibrations for communication, enabling signaling through solid materials rather than through air. Foraging behavior demonstrates sophisticated decision-making processes that optimize energy gain, while minimizing risks from predators and environmental hazards. Central place foragers such as bees and wasps must balance the energy costs of travel against the quality and quantity of resources at different locations. Search strategies may involve systematic scanning of areas, following chemical gradients, or responding to visual cues that indicate resource availability. Learning and memory enable insects to improve foraging efficiency by remembering profitable locations and avoiding unproductive areas. Navigation abilities in insects rival those of much larger animals, enabling precise movement through complex three-dimensional environments using multiple sensory cues and sophisticated information processing. Path integration allows insects to maintain awareness of their position relative to a reference point by continuously updating distance and direction information. Landmark navigation uses visual features to recognize specific locations and plan routes between them. Celestial navigation employs sun position, polarized light patterns, and stellar positions for long-distance orientation and migration. Learning and memory capabilities in insects enable behavioral flexibility and adaptation to changing environmental conditions, with some species demonstrating remarkable cognitive abilities despite their small nervous systems. Classical conditioning enables insects to associate stimuli with positive or negative outcomes, while operant conditioning allows them to modify behavior based on its consequences. Spatial learning enables navigation through complex environments, while social learning allows some species to acquire information from other individuals. The neurobiological mechanisms underlying insect learning provide insights into the fundamental basis of cognition. Social behavior in insects ranges from simple aggregations to complex eusocial societies, with elaborate division of labor and cooperative care of offspring. Eusocial species such as ants, bees, termites, and some wasps have evolved sophisticated social organization, including reproductive division of labor, overlapping generations, and cooperative brood care. The evolution of eusociality requires explanations for how individuals benefit from helping relatives reproduce rather than reproducing themselves. Kin selection theory provides a framework for understanding how genes for social behavior can spread through populations. The organization of insect societies involves complex systems of communication, division of labor, and conflict resolution that enable thousands of individuals to coordinate their activities effectively. Chemical communication systems in social insects include queen pheromones that regulate reproduction, recruitment pheromones that organize foraging, and recognition pheromones that maintain colony cohesion. Division of labor may be based on age, size, or genetic differences that predispose individuals to perform specific tasks. Conflict resolution mechanisms prevent disruptive competition within colonies while maintaining cooperative behavior. Mating behaviors and sexual selection have produced some of the most elaborate and diverse behavioral repertoires in the animal kingdom, with insects employing strategies ranging from resource defense to elaborate courtship displays. Males may compete directly for females through physical combat or territorial defense, or they may compete indirectly through sperm competition and mate guarding. Female choice often favors males with elaborate ornaments or complex courtship behaviors that indicate genetic quality or resource-holding potential. The diversity of insect mating systems reflects different ecological constraints and evolutionary pressures. Parental care in insects varies from simple egg-laying to complex provisioning and protection of offspring, with some species showing remarkable investment in offspring survival. Many species provision their eggs with nutrients or place them in protected locations with adequate food resources. Some insects build elaborate nests or brood chambers that protect developing offspring from predators and environmental hazards. Extended parental care may involve feeding offspring, defending them from threats, or teaching them important skills such as foraging techniques. Predator-prey interactions have shaped numerous aspects of insect behavior, leading to sophisticated strategies for both capturing prey and avoiding becoming prey themselves. Predatory insects may use ambush tactics, active pursuit, or trap construction to capture prey, while many species have evolved elaborate defensive behaviors including crypsis, aposematism, and mimicry. Anti-predator behaviors may involve freezing responses, escape behaviors, defensive secretions, or coordinated group defenses. The co-evolution of predatory and defensive strategies has contributed to the diversity of behavioral adaptations in insects. Migration and dispersal behaviors enable insects to exploit temporally and spatially variable resources while avoiding unfavorable environmental conditions. Some insects undertake remarkable long-distance migrations that span continents and multiple generations, requiring sophisticated navigation abilities and physiological adaptations for sustained flight. Dispersal strategies may involve active movement in response to environmental cues or passive transport by wind currents. The timing of migration and dispersal often coordinates with seasonal changes in resource availability or breeding opportunities. Habitat selection involves complex decision-making processes that evaluate multiple environmental factors to identify locations suitable for survival and reproduction. Insects may respond to visual cues such as plant color and shape, chemical cues indicating food quality or the presence of competitors, or physical factors such as temperature and humidity. Overposition site selection is particularly important as it determines the environment where offspring will develop. The evolution of habitat preferences reflects the balance between resource quality, predation risk, and competition intensity. Rhythmic behaviors and biological clocks enable insects to coordinate their activities with daily and seasonal cycles, optimizing timing of feeding, mating, and other activities. Circadian rhythms control the timing of emergence, foraging, and reproductive behaviors, while seasonal rhythms coordinate development and reproduction with favorable environmental conditions. The molecular mechanisms underlying biological clocks involve feedback loops of gene expression that maintain approximately 24-hour cycles even in constant environmental conditions. Understanding biological rhythms is important for predicting insect activity patterns and developing effective management strategies. Behavioral ecology examines how ecological factors influence the evolution and expression of insect behaviors, providing frameworks for understanding behavioral diversity and predicting responses to environmental change. Resource distribution affects foraging strategies and social organization, while predation pressure influences anti-predator behaviors and habitat selection. Competition for mates and resources shapes reproductive strategies and territorial behaviors. Climate and weather patterns affect activity levels and the timing of important life history events. The study of insect behavior continues to reveal new aspects of cognition, communication, and social organization, while providing insights into fundamental questions about behavior and evolution. Advanced techniques including video analysis, genetic manipulation, and neurobiological recording are enabling detailed investigation of behavioral mechanisms and their underlying neural basis. Understanding insect behavior has practical applications for pest management, conservation biology, and biomimetics, while contributing to our understanding of behavior and cognition in other animals including humans. Chapter 5: Diversity and Evolution. The diversity of insects surpasses that of any other group of animals, with over 1 million described species representing estimates suggests that several million more await discovery. This extraordinary richness reflects over 400 million years of evolutionary innovation that has produced insects adapted to virtually every conceivable ecological niche and lifestyle. Understanding insect diversity requires examining both the evolutionary processes that have generated this remarkable variety and the ecological factors that maintain it in contemporary ecosystems. The evolutionary origins of insects trace back to the early diversification of arthropods, with fossil evidence suggesting that hexapods diverged from other arthropod lineages during the Silurian period approximately 420 million years ago. Early insects were wingless, terrestrial arthropods that likely fed on decaying organic matter and early land plants. The evolution of wings during the Carboniferous period represented a revolutionary innovation that enabled insects to exploit three-dimensional space and dramatically expand their ecological opportunities. This aerial capability, combined with small size and high reproductive rates, set the stage for the explosive diversification that followed. The major orders of insects represent distinct evolutionary lineages that have diverged over hundreds of millions of years, each developing characteristic features that define their ecological roles and evolutionary success. The Coleoptera, or beetles, constitute the most species-rich order, with over 400,000 described species, demonstrating remarkable diversity in feeding strategies, body forms, and ecological adaptations. Their success stems from the evolution of hardened forewings that protect the flight wings and body, enabling exploitation of numerous microhabitats unavailable to other insects. Lepidoptera, including butterflies and moths, represent one of the most visible and ecologically important insect orders with over 180,000 described species. The evolution of the coiled proboscis enabled exploitation of floral nectar resources, leading to intimate co-evolutionary relationships with flowering plants. The larval stages, commonly known as caterpillars, have evolved diverse feeding strategies ranging from generalist herbivory to highly specialized relationships with particular plant species. Scale-covered wings enable complex visual displays and warning signals, while providing protection from predators. Hymenoptera, encompassing ants, bees, wasps, and their relatives, demonstrate remarkable diversity in social organization and ecological roles, with over 150,000 described species. The evolution of a narrow waist, connecting thorax and abdomen, enhanced mobility and enabled sophisticated behavioral repertoires. Many lineages have evolved eusocial organization, with complex division of labor and cooperative brood care. The evolution of specialized structures for pollen collection in bees has made them crucial pollinators of flowering plants worldwide. Diptera, the true flies, represent highly successful two-winged insects with over 125,000 described species that have colonized diverse habitats and feeding niches. The modification of hind wings into small balancing organs, called halteres, enhanced flight control and maneuverability. Many species have evolved specialized relationships with other organisms as parasites, parasitoids, or disease vectors. The complete metamorphosis of flies enables larvae and adults to exploit entirely different resources, reducing intraspecific competition while maximizing resource utilization. Hemiptera, including aphids, bugs, and cicadas, demonstrate the evolutionary success of piercing-sucking mouthparts, with over 100,000 described species. These insects have specialized for feeding on plant fluids, with some lineages evolving complex relationships with host plants including gall formation and virus transmission. Many species exhibit remarkable reproductive strategies, including parthenogenesis, and complex life cycles that alternate between sexual and asexual reproduction. Some hemipterans have evolved predatory lifestyles while retaining their characteristic mouthpart structure. The evolution of metamorphosis represents one of the most significant innovations in insect evolution, enabling the remarkable success of holometabolous orders that comprise over 80% of described insect species. Complete metamorphosis allows different life stages to specialize for different functions, with larvae focused on growth and resource acquisition, while adults concentrate on reproduction and dispersal. This division of labor reduces intraspecific competition and enables exploitation of diverse resources throughout the life cycle. The evolution of metamorphosis required complex developmental innovations including imaginal discs and sophisticated hormonal control systems. Adaptive radiation has repeatedly produced remarkable diversity within insect lineages when new ecological opportunities became available or when key innovations opened new adaptive zones. The diversification of herbivorous insects paralleled the evolution of flowering plants, with many insect lineages rapidly evolving to exploit new plant hosts and defensive strategies. Island archipelagos have provided natural laboratories for studying adaptive radiation, with endemic insect faunas evolving diverse forms adapted to local conditions and available niches. Co-evolution between insects and other organisms has driven much of insect diversification, while shaping the evolution of other life forms. Plant-insect interactions have resulted in remarkable specificity, with many insects specialized to feed on particular plant species or families. These relationships have driven the evolution of plant chemical defenses and insect detoxification systems in ongoing evolutionary arms races. Pollination mutualisms have shaped the evolution of both flowers and their insect visitors, while parasitic and predatory relationships have influenced the evolution of defensive strategies and life history traits. The role of sexual selection in insect evolution has produced some of the most elaborate morphological and behavioral traits in the animal kingdom. Male competition for mates has driven the evolution of weaponry, including enlarged mandibles, horns, and other fighting structures. Female choice has favored the evolution of complex courtship behaviors, elaborate ornaments, and nuptial gifts. The diversity of reproductive strategies reflects different solutions to the challenges of finding mates and ensuring reproductive success in various ecological contexts. Geographic patterns of insect diversity reveal the importance of climate, habitat heterogeneity, and evolutionary history in shaping species richness and endemism. Tropical regions support the highest insect diversity, reflecting favorable climatic conditions, habitat complexity, and long evolutionary history without major climatic disruptions. Mountain ranges and islands often support high levels of endemism due to geographic isolation that promotes speciation. Understanding these patterns is crucial for conservation efforts and predicting responses to environmental change. Speciation mechanisms in insects include both allopatric processes involving geographic isolation and sympatric processes occurring within continuously distributed populations. Host plant specialization has been particularly important in promoting speciation among herbivorous insects, with divergent adaptation to different plants leading to reproductive isolation. Behavioral differences in mating signals or preferences can lead to reproductive isolation even in the absence of geographic barriers. Polyploidy and other chromosomal changes can instantaneously create reproductive barriers in some insect lineages. The fossil record provides crucial insights into insect evolutionary history, while revealing the timing of major innovations and diversification events. Exceptionally preserved fossils from sites such as the Solnhofen limestone and Baltic amber provide detailed information about extinct insect faunas and their ecological relationships. Trace fossils including leaf damage patterns and coprolites reveal ancient feeding relationships and ecosystem interactions. The integration of fossil and molecular data is improving our understanding of insect phylogeny and evolutionary timing. Molecular phylogenetics has revolutionized our understanding of insect relationships and evolutionary history by providing independent evidence for testing morphology-based hypotheses. DNA sequence data has revealed unexpected relationships among insect orders while confirming some traditional groupings. The molecular clock approach estimates divergence times between lineages, revealing that major insect diversification began much earlier than previously thought. Comparative genomics is identifying the genetic changes underlying major evolutionary innovations including wing development, metamorphosis, and social behavior. Conservation of insect diversity faces numerous challenges including habitat destruction, climate change, pollution, and invasive species that threaten both individual species and entire ecosystems. Many insects have narrow habitat requirements or specialized ecological relationships that make them vulnerable to environmental changes. The small size and cryptic nature of many insects complicate conservation efforts by making population monitoring difficult. However, insects also show remarkable resilience and rapid evolutionary responses that may enable some species to adapt to changing conditions. The economic and ecological importance of insect diversity extends far beyond their intrinsic value, with insects providing essential ecosystem services including pollination, decomposition, and pest control while serving as food sources for countless other organisms. Agricultural systems depend heavily on insect pollinators, while natural ecosystems rely on insects for nutrient cycling and energy transfer. Understanding and maintaining insect diversity is crucial for ecosystem stability and human welfare. The study of insect diversity continues to reveal new species and evolutionary relationships, while highlighting the remarkable success of these animals in colonizing Earth's terrestrial environments. Climate change represents one of the most significant contemporary threats to insect diversity, with rising temperatures, altered precipitation patterns, and increased frequency of extreme weather events affecting insect distributions, life cycles, and ecological relationships. Some species may benefit from warmer temperatures and extended growing seasons, while others may face extinction as their specialized habitats disappear. Understanding how insects respond to climate change is crucial for predicting future biodiversity patterns and developing effective conservation strategies. The remarkable evolutionary flexibility that has enabled insects to dominate terrestrial ecosystems may also help them adapt to rapidly changing environmental conditions. Chapter 1: Plant Structure and Organization. Plants represent the foundation of terrestrial life, providing the energy and oxygen that sustain virtually all other living organisms on Earth. These remarkable organisms have evolved sophisticated structures and systems that enable them to capture sunlight, absorb water and nutrients from soil, and convert simple inorganic compounds into the complex organic molecules that fuel ecosystems worldwide. Understanding plant biology begins with examining their fundamental architecture and organization, which reflects millions of years of evolutionary adaptation to life at the interface between Earth and sky. The basic plant body plan exhibits a remarkable unity of design, despite the enormous diversity of plant forms found across different environments and taxonomic groups. This organization reflects the fundamental challenge faced by all terrestrial plants: the need to simultaneously access resources from two very different environments. Roots explore the dark, nutrient-rich soil environment to absorb water and minerals, while shoots extend into the aerial environment to capture light energy and exchange gases with the atmosphere. This division of labor between underground and above-ground organs has shaped plant evolution and continues to influence every aspect of plant biology. The modular construction of plants enables them to grow throughout their lives by adding new organs and tissues as needed, unlike animals which typically reach a determinate size. This indeterminate growth pattern allows plants to continuously explore their environment for resources while adjusting their body plan in response to changing conditions. New roots can grow toward water sources, while shoots can extend toward light and away from competitors. This growth flexibility represents one of the key innovations that has enabled plants to colonize diverse terrestrial environments. Roots serve as the foundation of plant life, anchoring plants in the substrate while providing essential functions including water and nutrient absorption, storage, and often reproduction. The root system begins with the primary root that emerges from the germinating seed, but this initial structure is quickly supplemented by lateral roots that branch extensively to explore the soil volume. Root architecture varies dramatically among species, with some plants developing deep taproots that access groundwater, while others produce shallow spreading root systems that efficiently capture surface moisture and nutrients. The cellular organization of roots reflects their specialized functions, with distinct tissues arranged in precise patterns that enable efficient resource absorption and transport. The root tip contains the apical meristem where new cells are continuously produced, protected by the root cap that facilitates penetration through soil particles. Behind the growing tip, the elongation zone enables rapid root extension, while the maturation zone develops specialized structures including root hairs that dramatically increase the surface area available for absorption. The internal organization includes the epidermis for protection and absorption, cortex for storage and transport, and vascular cylinder for long-distance transport of water and nutrients. Mycorrhizal associations represent one of the most important innovations in plant biology, involving intimate partnerships between plant roots and beneficial fungi that enhance nutrient absorption capabilities. These mutualistic relationships evolved early in plant evolution and are found in over 90% of plant species, indicating their fundamental importance for plant success. Arbuscular mycorrhizal fungi form branching structures within root cells that facilitate nutrient exchange, while ectomycorrhizal fungi form mantles around root tips and extend hyphal networks far into the surrounding soil. These fungal partners effectively expand the root system by orders of magnitude, while providing access to nutrients that would otherwise be unavailable. Stems provide the structural framework that positions leaves and reproductive organs, while serving as conduits for transporting materials between roots and shoots. The enormous diversity of stem forms reflects different evolutionary solutions to the challenges of mechanical support, resource transport, and environmental adaptation. Herbaceous stems remain soft and flexible throughout their lives, while woody stems develop secondary tissues that provide mechanical strength and enable plants to achieve great heights. Specialized stem modifications including rhizomes, stolons, and bulbs enable storage, reproduction, and survival through adverse conditions. The vascular system of plants represents a sophisticated network of specialized tissues that enable long-distance transport of water, nutrients, and organic compounds throughout the plant body. Xylem tissue transports water and dissolved minerals from roots to shoots through hollow tubes formed by dead cells with reinforced walls. The transpiration cohesion tension mechanism drives water movement by creating negative pressure as water evaporates from leaves, pulling water upward through continuous columns maintained by strong intermolecular forces. Phloem tissue transports organic compounds including sugars produced by photosynthesis from sources to sinks throughout the plant, using energy-dependent mechanisms that can move materials in multiple directions. Leaves represent the primary photosynthetic organs, where plants capture light energy and convert carbon dioxide into organic compounds that fuel all plant activities. Leaf architecture reflects optimization for light capture, while balancing competing demands including gas exchange, water conservation, and mechanical support. The flattened blade maximizes surface area for light interception, while the arrangement of tissues within the leaf creates specialized environments for different photosynthetic functions. Palisade mesophyll cells near the upper surface contain numerous chloroplasts optimally positioned for light capture, while spongy mesophyll cells facilitate gas exchange through extensive air spaces. The intricate structure of leaf stomata demonstrates the sophisticated control mechanisms that plants have evolved to regulate gas exchange and water loss. These microscopic pores consist of pairs of guard cells that can change shape to open or close the stomatal aperture in response to environmental conditions and internal signals. The ability to rapidly adjust stomatal opening enables plants to optimize carbon dioxide uptake for photosynthesis while minimizing water loss, a critical balance for survival in terrestrial environments. Stomatal density and distribution vary among species and can change in response to environmental conditions, demonstrating the plasticity of plant development. The cellular basis of plant structure reveals remarkable specialization that enables efficient function while maintaining structural integrity. Plant cells are distinguished by rigid cell walls composed primarily of cellulose that provide mechanical support and protection while allowing controlled expansion during growth. The cell wall structure includes multiple layers with different compositions and organizations that provide specific mechanical properties. Plasmodesmata create cytoplasmic connections between adjacent cells that enable communication and transport while maintaining cellular autonomy. The large central vacuole in mature plant cells serves multiple functions including mechanical support, storage, and waste management. Meristematic tissues represent the growth centers where new cells are continuously produced throughout the plant's life, enabling the indeterminate growth that characterizes plants. Apical meristems at root and shoot tips produce primary tissues that enable elongation and basic organ development, while lateral meristems, including the vascular cambium and cork cambium, produce secondary tissues that enable increased girth and protective functions. The organization of meristems reflects precise developmental control that maintains proper tissue relationships while allowing flexible responses to environmental conditions. Secondary growth in woody plants involves the production of secondary xylem wood and secondary phloem inner bark by the vascular cambium, enabling plants to achieve great size and longevity. The seasonal patterns of secondary growth create annual rings that provide records of environmental conditions and growth patterns throughout the plant's life. The mechanical properties of wood result from the orientation and chemical composition of cell walls, with different wood types optimized for support, transport, or storage functions. Cork cambium produces protective bark that replaces the epidermis in older stems and roots. Plant tissue systems demonstrate sophisticated organization that integrates multiple cell types into functional units capable of performing complex physiological processes. The dermal tissue system provides protection and regulates interactions with the environment through specialized structures including stomata, trichomes, and cuticles. The ground tissue system comprises the bulk of plant bodies and performs diverse functions including photosynthesis, storage, and support through different cell types with specialized structures and compositions. The vascular tissue system creates continuous transport networks that connect all parts of the plant body while providing mechanical support. The remarkable diversity of plant forms reflects evolutionary responses to different environmental challenges and opportunities. Desert plants have evolved succulent tissues for water storage and modified leaves that reduce water loss while maintaining photosynthetic capacity. Aquatic plants show reduced structural tissues and specialized features for underwater life, including modified leaves and stems. Climbing plants have evolved various attachment mechanisms and flexible stems that enable them to reach light without investing heavily in support tissues. These adaptations demonstrate the extraordinary plasticity of the basic plant body plan. Plant architecture continues to inspire human engineering and design, providing models for efficient structures and systems. The branching patterns of roots and shoots optimize resource capture while minimizing material investment, principles that inform modern network design and architectural planning. The hierarchical organization of plant tissues demonstrates how complex functions can emerge from simple cellular components, insights that guide biomimetic material development. Understanding plant structure and organization provides the foundation for appreciating all other aspects of plant biology, while revealing the elegant solutions that evolution has produced for the challenges of terrestrial life. Chapter 2: Photosynthesis and Plant Metabolism. Photosynthesis stands as perhaps the most important biological process on Earth, transforming light energy into chemical energy while simultaneously converting atmospheric carbon dioxide into organic compounds that form the foundation of virtually all food webs. This remarkable process enables plants to serve as primary producers that capture and channel solar energy into ecosystems, supporting the entire web of life on our planet. Understanding photosynthesis reveals the sophisticated molecular machinery that plants have evolved to harvest light energy, while managing the complex chemistry required to build organic molecules from simple inorganic compounds. The fundamental equation of photosynthesis appears deceptively simple, combining carbon dioxide and water in the presence of light to produce glucose and oxygen. Yet this process involves some of the most complex biochemical machinery known to science. The conversion of light energy into chemical bonds requires precise coordination between light-capturing systems, electron transport chains and carbon-fixing enzymes operating in specialized cellular compartments. The efficiency and elegance of photosynthetic systems have inspired decades of research aimed at understanding and mimicking these natural solar energy conversion systems. Chloroplasts represent the cellular powerhouses where photosynthesis occurs, containing sophisticated membrane systems and molecular machinery optimized for light capture and energy conversion. These organelles evolved from ancient cyanobacteria through endosymbiotic relationships, retaining their own genetic systems while becoming integrated into plant cellular functions. The internal structure of chloroplasts includes an elaborate thylakoid membrane system where light reactions occur, surrounded by the stroma where carbon fixation takes place. This compartmentalization enables precise control over the chemical environments required for different aspects of photosynthesis. The light-dependent reactions of photosynthesis demonstrate remarkable molecular engineering that captures photons and converts their energy into chemical forms that can power cellular processes. Photosystem 2 initiates the process by using light energy to split water molecules, releasing electrons, protons, and oxygen, while generating the oxygen that makes aerobic life possible. The electrons pass through an electron transport chain that includes photosystem I, creating a proton gradient across thylakoid membranes that drives ATP synthesis. This photochemical machinery operates with quantum efficiency, approaching theoretical limits while managing the potentially damaging effects of high-energy photons. Chlorophyll and accessory pigments work together to create sophisticated light harvesting systems that capture photons across broad spectral ranges while protecting cellular components from photodamage. Chlorophyll A serves as the primary photosynthetic pigment, while chlorophyll B, carotenoids and other accessory pigments extend the range of usable wavelengths and provide photoprotective functions. The arrangement of pigments in antenna complexes creates efficient energy transfer systems that funnel captured light energy to reaction centers where photochemistry occurs. The diversity of photosynthetic pigments enables plants to adapt to different light environments while maintaining efficient energy capture. The Calvin cycle represents the biochemical pathway where atmospheric carbon dioxide is incorporated into organic molecules, through a series of enzyme-catalyzed reactions that require the ATP and NADPH produced by light reactions. Ribulose bisphosphate carboxylase oxygenase, RU-BIS-CO, serves as the key enzyme that catalyzes carbon fixation, despite being one of the most abundant proteins on Earth due to its relatively low catalytic efficiency. The cycle involves 3 phases: carbon fixation, reduction, and regeneration, with complex regulation ensuring that carbon flow matches the availability of energy and reducing power from light reactions. Alternative photosynthetic pathways have evolved to address the limitations of C3 photosynthesis in specific environmental conditions, demonstrating the evolutionary flexibility of plant metabolism. C4 photosynthesis involves spatial separation of initial carbon fixation and the Calvin cycle, concentrating carbon dioxide around RU-BIS-CO and reducing photorespiration in hot, dry environments. Crassulacean acid metabolism (CAM) achieves similar benefits through temporal separation, with carbon dioxide fixation occurring at night when stomata can open with minimal water loss. These adaptations enable plants to maintain photosynthetic productivity under conditions where C3 photosynthesis would be inefficient. Photorespiration represents a metabolic process that can reduce photosynthetic efficiency when RU-BIS-CO reacts with oxygen instead of carbon dioxide, particularly under conditions of high temperature, low carbon dioxide, or high oxygen concentrations. This process involves coordination between chloroplasts, peroxisomes, and mitochondria to salvage carbon and minimize energy losses, though it still represents a significant metabolic cost. The evolution of C4 and CAM photosynthesis can be viewed as responses to photorespiration, while ongoing research explores ways to engineer plants with reduced photorespiratory losses. Cellular respiration in plants operates continuously to provide the energy and carbon skeletons needed for growth, maintenance, and reproduction, demonstrating that plants, like all living organisms, must break down organic molecules to fuel their metabolism. Plant respiration involves the same basic pathways found in other organisms, including glycolysis, the citric acid cycle, and electron transport, but occurs in different cellular compartments and shows unique features related to plant-specific metabolism. The balance between photosynthetic carbon gain and respiratory carbon loss determines net carbon accumulation and plant growth rates. The integration of photosynthesis and respiration creates complex metabolic networks that enable plants to respond to changing environmental conditions while maintaining essential cellular functions. During daylight hours, photosynthesis typically exceeds respiration, resulting in net carbon dioxide fixation and organic molecule accumulation. During darkness, respiration continues while photosynthesis ceases, drawing on stored carbohydrates to maintain cellular activities. The coordination between these processes involves sophisticated regulatory mechanisms that sense cellular energy status and environmental conditions. Carbohydrate metabolism in plants involves complex pathways for synthesizing, modifying, and storing various sugar molecules that serve as energy sources, structural components, and signaling molecules. Starch serves as the primary storage carbohydrate in most plants, accumulating in chloroplasts during photosynthesis and being mobilized during periods of high energy demand or limited photosynthesis. Sucrose functions as the primary transport sugar, moving photosynthetic products from source tissues to sink tissues throughout the plant. Cellulose synthesis creates the structural carbohydrates that form cell walls and provide mechanical support. Lipid metabolism in plants includes the synthesis of membrane lipids, storage oils, and specialized compounds including waxes and signaling molecules that serve diverse functions in plant physiology and ecology. Plant oils represent highly concentrated energy storage forms that are particularly important in seeds, where efficient energy storage is critical for successful germination and establishment. Cuticular waxes provide waterproofing and protection for aerial plant surfaces, while specialized lipids serve signaling functions in plant development and defense responses. Nitrogen metabolism links plant nutrition with protein synthesis and other essential cellular processes, requiring the incorporation of nitrogen from inorganic sources into organic compounds. Plants absorb nitrogen primarily as nitrate or ammonium from soil, then reduce and incorporate it into amino acids through pathways that require significant energy investment. Nitrogen availability often limits plant growth, making nitrogen metabolism a critical determinant of plant productivity and ecosystem dynamics. The efficiency of nitrogen use varies among species and can be modified through breeding and genetic engineering approaches. Secondary metabolism produces the enormous diversity of specialized compounds that plants use for defense, signaling, and environmental adaptation, contributing to the chemical diversity that makes plants valuable sources of pharmaceuticals, materials, and other useful compounds. These pathways often involve modifications of primary metabolic products to create alkaloids, phenolics, terpenoids, and other compound classes with specific biological activities. Secondary metabolism demonstrates remarkable creativity in molecular design while providing insights into the evolutionary pressures that have shaped plant chemistry. Metabolic regulation in plants involves complex networks of feedback controls, allosteric regulation, and transcriptional control that coordinate metabolic activities with cellular needs and environmental conditions. Light sensing systems integrate information about light quality, quantity, and duration to coordinate photosynthetic metabolism with environmental conditions. Sugar sensing mechanisms adjust metabolic activities based on cellular energy status, while stress sensing systems modify metabolism in response to environmental challenges including drought, temperature extremes, and pathogen attack. Photosynthetic acclimation demonstrates the remarkable flexibility of plant metabolism in responding to changing light environments, with plants adjusting their photosynthetic apparatus to optimize energy capture and utilization under different conditions. High light acclimation involves increasing the capacity for light utilization, while enhancing photoprotective mechanisms that prevent damage from excess light energy. Low light acclimation involves increasing light harvesting capacity, while minimizing respiratory costs that would compete with limited photosynthetic energy gain. These acclimation responses involve coordinated changes in gene expression, protein synthesis, and metabolic regulation. Understanding plant metabolism continues to provide insights into fundamental biological processes while revealing opportunities for crop improvement and biotechnology applications. Metabolic engineering approaches seek to enhance photosynthetic efficiency, improve stress tolerance, and increase the production of valuable compounds by modifying metabolic pathways. Research into artificial photosynthesis aims to harness the principles of plant energy conversion for renewable energy applications. The study of plant metabolism demonstrates the sophisticated chemistry that underlies plant life while highlighting the central role of plants in global energy and carbon cycles. Chapter 3: Growth, Development, and Reproduction. Plant growth and development represent dynamic processes that continue throughout the plant's life, enabling continuous adaptation to changing environmental conditions while coordinating the formation of new organs and tissues. Unlike animals, which typically reach a determinate size and cease growing, plants exhibit indeterminate growth that allows them to continuously explore their environment and modify their architecture in response to local conditions. This growth plasticity, combined with sophisticated developmental programs, enables plants to optimize their form and function for survival and reproduction in diverse environments. The fundamental mechanisms of plant development involve complex interactions between genetic programs and environmental signals that guide cell division, differentiation, and organ formation. Plant development begins with embryogenesis within the seed, where basic body plan elements including root and shoot apical meristems are established. These meristematic regions contain populations of undifferentiated cells that retain the capacity for continuous division and differentiation throughout the plant's life. The precise regulation of meristematic activity determines plant size, shape, and organ production patterns. Cell division in plants occurs in specific patterns that contribute to overall plant architecture while maintaining proper tissue organization and function. Unlike animal cells, plant cells cannot migrate after division due to their rigid cell walls, making the plane and timing of cell division critical for proper development. Oriented cell divisions create specific tissue arrangements and contribute to organ shape, while the coordination of cell division rates among different tissues maintains proportional relationships during growth. Molecular mechanisms including microtubule arrays and cell cycle checkpoints ensure proper division orientation and timing. Cell expansion represents the primary mechanism for plant growth, with individual cells increasing in volume by up to 1,000-fold through controlled wall loosening and turgor pressure. This process requires precise coordination between wall synthesis, wall loosening, and osmotic regulation to maintain cellular integrity while enabling dramatic size increases. The direction of cell expansion is controlled by the orientation of cellulose microfibrils in the cell wall, which in turn is influenced by cortical microtubule arrays that guide cellulose synthesis. Hormonal signals coordinate expansion among different cells and tissues to produce coherent organ growth. Differentiation transforms undifferentiated meristematic cells into specialized cell types with specific structures and functions, creating the tissue diversity that enables complex plant functions. This process involves progressive changes in gene expression that determine cell fate while maintaining cellular viability and function. Differentiation patterns are influenced by positional information that cells receive based on their location within developing organs, as well as by hormonal signals that coordinate development across the entire plant. The timing of differentiation must be carefully controlled to ensure that specialized cells develop when and where they are needed. Plant hormones serve as chemical messengers that coordinate growth and development by regulating cell division, expansion, and differentiation throughout the plant body. Auxin plays central roles in cell elongation, tissue differentiation, and organ development, while coordinating responses to environmental stimuli including light and gravity. Gibberellins promote stem elongation and seed germination, while influencing flowering and fruit development. Cytokinins stimulate cell division and delay senescence, while affecting shoot development and nutrient allocation. The interactions among different hormones create complex regulatory networks that fine-tune developmental processes. Gravitropism demonstrates how plants sense and respond to environmental stimuli through sophisticated sensory systems that convert physical signals into developmental responses. Root gravitropism ensures that roots grow downward toward water and nutrients, while shoot gravitropism directs stem growth upward toward light. The sensing mechanism involves dense starch-containing organelles called statoliths that settle within specialized cells in response to gravity, triggering signal transduction pathways that lead to asymmetric growth responses. This system enables plants to maintain proper orientation despite changes in position or environmental conditions. Phototropism illustrates plant responses to light direction that optimize light capture for photosynthesis while demonstrating the integration of environmental sensing with growth control. Phototropic responses involve photoreceptors that detect light quality and direction, triggering signaling pathways that result in asymmetric growth and organ reorientation. Blue light photoreceptors, called phototropins, initiate phototropic responses by activating kinase cascades that ultimately affect auxin transport and distribution. These responses enable plants to track the sun's movement and optimize light interception throughout the day. Root development involves complex programs that generate diverse root types and arrangements, suited for different environmental conditions and functional requirements. Primary root development establishes the initial root system architecture, while lateral root formation creates branching patterns that maximize soil exploration. Root hair development increases surface area for absorption, while specialized roots including storage roots, prop roots, and aerial roots serve additional functions. Root development responds to soil moisture, nutrient distribution, and mechanical impedance through sophisticated sensing and signaling systems. Shoot development coordinates the formation of stems, leaves, and reproductive organs through programs that balance light capture, mechanical support, and reproductive success. Shoot apical meristems maintain populations of stem cells while producing the primordia that develop into leaves and lateral shoots. Phyllotaxis, the arrangement of leaves around stems, follows mathematical patterns that optimize light interception while minimizing self-shading. Branching patterns determine plant architecture and influence resource allocation between growth and reproduction. Leaf development involves complex morphogenetic processes that create the diverse leaf shapes and structures found across plant species. Early development establishes basic leaf identity and polarity, while subsequent growth creates the final leaf form through coordinated cell division and expansion. Leaf margin development can produce smooth serrated or lobed edges depending on species-specific programs and environmental conditions. Venation patterns develop through complex interactions between hormone gradients and mechanical forces that create efficient transport networks. Flower development represents one of the most complex and precisely controlled developmental programs in plants, involving the transformation of vegetative shoot apical meristems into reproductive structures with specific arrangements of sepals, petals, stamens, and carpels. The ABC model of flower development describes how different combinations of homeotic genes specify the identity of different floral organs, with precise spatial and temporal regulation ensuring proper organ formation. Flower development must be coordinated with environmental conditions and plant physiological status to ensure successful reproduction. Flowering time regulation integrates information about day length, temperature, age, and nutritional status to ensure that reproduction occurs under favorable conditions. Photoperiodic responses enable plants to time flowering with seasons by measuring day length through complex molecular clocks and light sensing systems. Vernalization responses require exposure to cold temperatures before flowering can occur, ensuring that reproduction is delayed until after winter. Age-related flowering involves the accumulation of developmental signals that eventually overcome vegetative growth programs. Fruit and seed development involve maternal tissues that support developing embryos while creating dispersal structures that enhance reproductive success. Fruit development often involves dramatic changes in size, color, and chemical composition that attract dispersal agents while protecting developing seeds. Seed development creates storage organs containing embryos and nutritional reserves necessary for successful germination and establishment. The coordination between fruit and seed development ensures that both components mature simultaneously for optimal reproductive success. Senescence represents programmed aging processes that affect individual organs or entire plants, involving controlled breakdown of cellular components and reallocation of nutrients. Leaf senescence enables plants to recover valuable nutrients from aging leaves while reducing maintenance costs during periods of environmental stress. The timing of senescence is controlled by environmental factors, including day length and temperature, as well as by internal factors including hormonal signals and nutritional status. Understanding senescence has important applications for agriculture and horticulture. Vegetative reproduction enables plants to reproduce asexually through various mechanisms including fragmentation, specialized reproductive organs, and apomixis. These strategies allow successful genotypes to proliferate rapidly, while avoiding the costs and uncertainties of sexual reproduction. Vegetative reproduction is particularly common in environments where sexual reproduction is difficult or unreliable, such as Arctic regions or disturbed habitats. Many economically important plants rely on vegetative reproduction for propagation and crop production. Developmental plasticity enables plants to modify their development in response to environmental conditions, demonstrating remarkable flexibility in growth and form. Phenotypic plasticity allows single genotypes to produce different phenotypes under different environmental conditions, optimizing fitness across variable environments. This plasticity involves environmental sensing systems that detect relevant conditions and signaling pathways that modify developmental programs accordingly. Understanding developmental plasticity provides insights into plant adaptation and evolution, while informing agricultural and conservation strategies. Modern research techniques including genomics, molecular biology, and advanced imaging are revealing new details about plant growth and development, while providing tools for crop improvement and biotechnology applications. Genetic engineering approaches enable modification of developmental programs to improve crop yields, stress tolerance, and nutritional content. Understanding plant development continues to provide insights into fundamental biological processes while revealing the remarkable complexity and sophistication of plant life. Chapter 4: Plant Interactions and Ecology. Plants exist within complex webs of ecological relationships that influence every aspect of their biology, from individual physiology to ecosystem-level processes. These interactions span the full spectrum from beneficial mutualisms to antagonistic conflicts, creating selective pressures that have shaped plant evolution, while determining the structure and function of terrestrial ecosystems. Understanding plant ecology requires examining how plants interact with their physical environment, other organisms, and each other, while recognizing that these relationships operate across multiple spatial and temporal scales. Competition among plants for essential resources including light, water, and nutrients represents one of the most fundamental ecological interactions that shapes plant communities and drives evolutionary adaptation. Above-ground competition for light creates vertical stratification in plant communities, with different species adapted for different light environments from bright canopy conditions to deeply shaded understory. This competition has driven the evolution of various strategies including rapid height growth, efficient light capture, and shade tolerance. Below-ground competition for water and nutrients involves complex root interactions that are often invisible but equally important for determining competitive outcomes. Resource partitioning enables multiple plant species to coexist by utilizing different resources or the same resources in different ways, reducing direct competition while maximizing community diversity. Temporal partitioning involves species with different seasonal activity patterns, such as spring ephemeral wildflowers that complete their life cycles before tree canopies fully develop. Spatial partitioning includes species with different rooting depths or growth forms that access different resource pools. Chemical partitioning involves species that utilize different forms of nutrients, or that can access resources unavailable to competitors. Plant-herbivore interactions represent dynamic relationships where plants serve as food sources for various animals, while evolving defenses that reduce herbivory damage. These interactions have driven remarkable diversification in both plant defensive strategies and herbivore adaptations, creating co-evolutionary arms races that continue to shape ecological communities. Plant defenses include physical barriers such as thorns and tough leaves, chemical compounds that deter or poison herbivores, and induced responses that increase defensive capabilities when damage occurs. The evolution of these defenses reflects the ongoing selective pressure exerted by herbivorous animals. Chemical defense systems in plants produce an enormous diversity of secondary metabolites that serve protective functions, while contributing to the chemical diversity that makes plants valuable sources of pharmaceuticals and other useful compounds. Alkaloids, phenolics, terpenoids and other compound classes provide defense against herbivores, pathogens, and competitors through various mechanisms including toxicity, deterrence, and interference with biological processes. Many defensive compounds are produced constitutively as standing defenses, while others are induced only when plants detect damage or threat signals. Induced defenses demonstrate sophisticated plant responses to herbivory that can increase resistance while minimizing the metabolic costs of defense when threats are absent. These responses can include the production of defensive chemicals, changes in nutritional quality, or the release of volatile compounds that attract predators of herbivores. Systemic-induced responses can protect undamaged plant parts from future attack, while communication between plants can warn neighbors of impending danger. The sophistication of these responses reveals that plants are far from passive victims of herbivory. Pollination represents one of the most important mutualistic relationships in terrestrial ecosystems, involving interactions between flowering plants and various animal pollinators that benefit both partners. Plants provide resources including nectar and pollen to attract pollinators, while animals transfer pollen between flowers and enable sexual reproduction. These relationships have driven remarkable co-evolutionary diversification, with different plant species specialized for different pollinator types including bees, butterflies, birds, and bats. The precision of some pollination relationships demonstrates extreme co-evolutionary adaptation. Seed dispersal interactions enable plants to move their offspring away from parent plants, while providing benefits to animal dispersers, creating another important category of mutualistic relationships. Fleshy fruits provide nutritional rewards to animals that transport seeds to new locations, while various seed modifications including hooks, wings, and explosive mechanisms enable dispersal through different vectors. Long-distance dispersal is particularly important for colonizing new habitats and maintaining genetic connectivity between plant populations. The diversity of dispersal mechanisms reflects the importance of this process for plant reproductive success. Plant-microbe interactions encompass a vast array of relationships ranging from beneficial symbioses to pathogenic infections, with microorganisms playing crucial roles in plant nutrition, health, and stress tolerance. Rhizobial bacteria form nitrogen-fixing nodules on legume roots, converting atmospheric nitrogen into forms that plants can utilize while receiving carbon compounds in return. This relationship enables legumes to thrive in nitrogen-poor soils, while contributing nitrogen to ecosystems through decomposition and root exudation. The specificity of rhizobial relationships demonstrates precise co-evolutionary adaptation. Mycorrhizal associations between plant roots and beneficial fungi represent perhaps the most widespread and important mutualistic relationships in terrestrial ecosystems, with over 90% of plant species participating in these partnerships. Arbuscular mycorrhizal fungi enhance plant access to phosphorus and other nutrients while receiving carbon compounds from photosynthesis. Ectomycorrhizal fungi form extensive hyphal networks that connect multiple plants and facilitate nutrient and information exchange. These relationships are so fundamental that many plants cannot survive without their fungal partners. Plant pathogens including fungi, bacteria, viruses, and other microorganisms cause diseases that significantly impact plant health and survival while driving the evolution of plant immune systems. Plant pathogen recognition involves sophisticated surveillance systems that detect pathogen-associated molecular patterns and initiate defensive responses. The hypersensitive response involves programmed cell death that limits pathogen spread, while systemic acquired resistance provides protection against future infections. The molecular arms race between plants and pathogens continues to drive evolutionary innovation in both partners. Allelopathy involves the production of chemical compounds by plants that influence the growth and survival of neighboring plants, representing a form of chemical warfare that can significantly affect plant community composition. Allelopathic compounds can inhibit seed germination, reduce growth rates, or increase susceptibility to other stresses, providing competitive advantages to species that produce them. These interactions are particularly important in communities where direct resource competition is limited, such as arid environments where water stress makes direct competition less intense. Plant community assembly involves complex processes that determine which species coexist in particular locations and how their relative abundances change over time. Environmental filtering selects for species with traits suited for local abiotic conditions, while biotic interactions including competition and facilitation influence community composition. Dispersal limitation affects which species can reach suitable habitats, while historical factors including disturbance and chance events can have lasting effects on community structure. Understanding these processes is crucial for predicting vegetation responses to environmental change. Succession involves predictable changes in plant community composition following disturbances, demonstrating how ecological interactions and environmental constraints interact to shape vegetation development. Primary succession occurs on newly exposed substrates lacking soil, beginning with pioneer species that can tolerate harsh conditions and gradually developing into more complex communities. Secondary succession follows disturbances that leave soil intact, typically proceeding more rapidly than primary succession. The mechanisms driving succession include changing resource availability, facilitation among species, and shifts in competitive hierarchies. Climate change is altering plant ecological interactions through changes in temperature, precipitation, and atmospheric carbon dioxide concentrations that affect plant physiology, phenology, and geographic distributions. Rising temperatures are shifting species ranges toward poles and higher elevations, while changing precipitation patterns are altering competitive relationships and community composition. Elevated carbon dioxide can enhance plant growth, but may also affect plant-herbivore interactions by changing tissue chemistry. Understanding these changes is crucial for predicting ecosystem responses to continued climate change. Invasive species represent non-native plants that establish and spread in new environments, often causing significant ecological and economic impacts through competition with native species and alteration of ecosystem processes. Successful invaders often possess traits that enable rapid growth and reproduction in new environments, while native species may lack defenses against novel competitors. Biological invasions provide natural experiments for understanding ecological processes, while highlighting the importance of biogeographic barriers in maintaining ecosystem integrity. Conservation biology applies ecological principles to protect plant species and communities from threats including habitat loss, climate change, and invasive species. Ex situ conservation involves maintaining plant genetic diversity in botanical gardens and seed banks, while in situ conservation focuses on protecting natural habitats and ecological processes. Restoration ecology seeks to repair damaged ecosystems by re-establishing native plant communities and ecological functions. Understanding plant ecological interactions is essential for developing effective conservation strategies. Ecosystem services provided by plants include not only obvious benefits such as food and timber production, but also critical regulatory services including climate regulation, water cycling, and soil stabilization. Plants influence local and global climate through evapotranspiration and carbon sequestration, while root systems prevent soil erosion and maintain watershed function. The economic value of these services often exceeds the value of direct plant products, highlighting the importance of maintaining healthy plant communities for human welfare. Agricultural ecology applies ecological principles to crop production systems, recognizing that sustainable agriculture requires understanding and managing ecological interactions within and around agricultural landscapes. Crop rotation and polyculture systems can enhance soil health and reduce pest problems while maintaining productivity. Integrated pest management combines biological, cultural and chemical approaches to minimize pest damage while reducing environmental impacts. Agroecological approaches seek to design agricultural systems that mimic natural ecosystem functions while producing food and other needed products. The study of plant interactions and ecology continues to reveal new aspects of plant biology, while providing insights necessary for addressing environmental challenges including climate change, biodiversity loss, and sustainable resource management. Advanced techniques including molecular ecology, remote sensing, and large-scale experiments are enabling new discoveries about plant ecological relationships while informing conservation and management strategies. Understanding plant ecology remains essential for maintaining the ecological foundation that supports all terrestrial life. Chapter 5: Plant Evolution and Diversity. The evolutionary history of plants spans over 500 million years, during which time these remarkable organisms have transformed the terrestrial environment while diversifying into the hundreds of thousands of species that dominate land-based ecosystems today. This evolutionary journey represents one of the most significant transitions in the history of life on Earth, involving the colonization of land by aquatic ancestors and the subsequent evolution of increasingly sophisticated adaptations for terrestrial life. Understanding plant evolution reveals the innovations that enabled plants to thrive on land while generating the diversity that characterizes modern flora. The transition from aquatic to terrestrial environments required fundamental innovations that enabled plants to survive in an environment lacking the buoyant support and consistent moisture availability of aquatic habitats. Early land plants faced challenges including desiccation, mechanical support, nutrient acquisition, and reproduction in the absence of water. The evolution of waxy cuticles provided protection against water loss, while the development of simple vascular tissues enabled transport of water and nutrients. These early innovations laid the foundation for all subsequent plant evolution and enabled the colonization of terrestrial environments. The earliest land plants, represented today by bryophytes including mosses, liverworts, and hornworts, demonstrate many of the fundamental adaptations necessary for terrestrial life, while retaining characteristics that reflect their aquatic ancestry. These plants lack true vascular tissues and remain dependent on external water for reproduction, limiting their size and distribution to moist environments. However, they possess innovations including waxy cuticles, specialized reproductive structures, and simple conducting tissues that enabled successful colonization of land. Bryophytes continue to play important ecological roles as pioneer species and in nutrient cycling. The evolution of vascular tissues marked a crucial innovation that enabled plants to achieve greater size and complexity while accessing new ecological opportunities. The development of xylem tissue for water transport and phloem tissue for organic compound transport created internal transport systems that could support larger plant bodies while enabling specialization of different plant parts for different functions. Early vascular plants including club mosses, horsetails, and ferns achieved considerable success and dominated many terrestrial environments before the rise of seed plants. Seed evolution represents perhaps the most significant innovation in plant evolutionary history, enabling reproduction without dependence on external water, while providing embryos with nutrients and protection during establishment. Seeds combine the benefits of sexual reproduction with improved dispersal capabilities and enhanced survival during adverse conditions. The evolution of seeds required coordinated innovations including ovules, pollen, and specialized reproductive structures that enabled internal fertilization and embryo development. This innovation opened entirely new evolutionary possibilities and enabled seed plants to dominate terrestrial environments. The diversification of seed plants involved two major lineages that employed different strategies for reproduction and development. Gymnosperms including conifers, cycads, and ginkgos produce seeds on specialized structures without enclosing them in fruits. These plants dominated terrestrial environments for millions of years and continue to be important in many ecosystems, particularly in temperate and boreal regions. The success of gymnosperms reflects adaptations including efficient water transport, reduced nutritional requirements, and tolerance of environmental stress. Angiosperms, or flowering plants, represent the most recent and successful major innovation in plant evolution, diversifying explosively to become the dominant vegetation in most terrestrial environments. The evolution of flowers and fruits enabled more efficient reproduction and seed dispersal, while facilitating complex relationships with animal pollinators and dispersers. The closed carpel that encloses developing seeds provides additional protection and enables precise control over fertilization and seed development. These innovations, combined with more efficient vascular systems and diverse growth forms, enabled angiosperms to colonize virtually every terrestrial habitat. The co-evolution of flowering plants and their animal associates has driven much of the diversification observed in both groups, creating intricate relationships that influence ecosystem structure and function. The evolution of flowers reflects adaptation to different pollinator types, with flower shape, color, scent, and rewards optimized for specific animal visitors. Similarly, fruit evolution reflects adaptation to different dispersal agents, with fruit characteristics matched to the sensory capabilities and dietary preferences of target dispersers. These relationships demonstrate the importance of biotic interactions in driving evolutionary innovation. Polyploidy, the presence of multiple complete sets of chromosomes, has played a particularly important role in plant evolution and continues to be a major source of plant diversity. Polyploidy can arise through various mechanisms including unreduced gamete formation and hybridization between species, often resulting in instant reproductive isolation and the potential for rapid speciation. Many economically important plants including wheat, cotton, and strawberries are polyploids, while polyploidy is especially common in certain plant families and geographic regions. The tolerance of plants for polyploidy reflects their developmental flexibility and contributes to their evolutionary success. Adaptive radiation has repeatedly produced remarkable plant diversity when new ecological opportunities became available or when key innovations opened new adaptive zones. The colonization of islands often leads to rapid diversification, as plants adapt to novel environmental conditions and reduced competition. The Hawaiian silversword alliance exemplifies such radiation, with a single ancestral species diversifying into dozens of species with dramatically different growth forms and ecological adaptations. These radiations provide insights into the factors that promote diversification and the speed with which evolution can operate. Molecular phylogenetics has revolutionized our understanding of plant evolutionary relationships by providing independent evidence for testing morphology-based hypotheses about plant classification and evolution. DNA sequence data has revealed unexpected relationships among plant groups while confirming some traditional classifications. The molecular clock approach has enabled estimation of divergence times between lineages, revealing that major plant evolutionary innovations occurred much earlier than suggested by the fossil record. Comparative genomics is identifying the genetic changes underlying major evolutionary transitions, including the evolution of flowers, seeds, and vascular tissues. The fossil record provides crucial evidence for plant evolutionary history, while revealing the timing of major innovations and the environmental contexts in which they occurred. Exceptionally preserved fossils from sites including the Rhynie chert and coal deposits provide detailed information about ancient plant anatomy and ecology. Trace fossils including fossilized pollen and spores extend the record of plant evolution beyond what can be learned from structural remains. The integration of fossil and molecular data is improving our understanding of plant phylogeny and the environmental factors that influenced plant evolution. Convergent evolution has repeatedly produced similar solutions to environmental challenges in unrelated plant lineages, demonstrating the constraints imposed by physical and biological factors on plant form and function. Succulent growth forms have evolved independently in numerous plant families as adaptations to arid environments, while climbing habits have evolved repeatedly as solutions to competition for light. These examples show that similar selective pressures can produce similar evolutionary outcomes, while highlighting the importance of environmental factors in shaping evolution. Extinction events have profoundly influenced plant evolutionary history by eliminating major plant groups and creating opportunities for surviving lineages to diversify and fill vacant ecological roles. The end Permian extinction eliminated many gymnosperm groups and created opportunities for the rise of modern conifer families. The Cretaceous-Paleogene extinction that eliminated non-avian dinosaurs also affected plant communities and may have influenced the continued diversification of flowering plants. Understanding extinction and recovery patterns provides insights into plant evolutionary dynamics and resilience. Geographic patterns of plant diversity reflect the combined influences of evolutionary history, environmental factors, and ecological processes that determine where different plant groups can survive and reproduce. Tropical regions support the highest plant diversity, reflecting favorable climatic conditions, environmental stability, and long evolutionary history without major climatic disruptions. Mountain regions often support high levels of endemism due to geographic isolation and diverse environmental conditions that promote speciation. Island floras typically show high levels of endemism, but reduced diversity compared to mainland areas of similar size. Human activities have increasingly influenced plant evolution through habitat modification, species introductions, and selective breeding that have altered natural evolutionary processes. Agricultural plant breeding has produced remarkable morphological and physiological changes in crop species, demonstrating the potential for rapid evolutionary change under strong selection. The introduction of exotic species has created novel evolutionary pressures and opportunities, while sometimes leading to hybridization between previously isolated species. Climate change is altering selective pressures worldwide and may drive rapid evolutionary responses in plant populations. Conservation of plant diversity faces numerous challenges, including habitat destruction, climate change, and invasive species that threaten both individual species and evolutionary processes. Many plant species have narrow geographic ranges or specialized habitat requirements that make them vulnerable to environmental changes. Ex situ conservation efforts, including botanical gardens and seed banks, attempt to preserve plant genetic diversity outside natural habitats, while in situ conservation focuses on protecting natural populations and evolutionary processes. Understanding plant evolutionary history and processes is crucial for developing effective conservation strategies. The future of plant evolution will likely be influenced by continued human activities and environmental changes that create new selective pressures and evolutionary opportunities. Climate change may drive rapid evolutionary responses as plants adapt to changing temperature and precipitation patterns. Continued habitat fragmentation may reduce population sizes and limit gene flow, affecting evolutionary potential. However, the remarkable evolutionary flexibility that has enabled plants to colonize virtually every terrestrial environment suggests that many plant lineages may successfully adapt to future environmental changes. Modern research techniques, including genomics, phylogenetics, and experimental evolution, are revealing new aspects of plant evolutionary history while providing tools for understanding evolutionary processes and predicting future changes. Whole genome sequencing is identifying the genetic basis of major evolutionary innovations, while revealing patterns of genome evolution across plant lineages. Experimental evolution studies, using model organisms, are testing evolutionary hypotheses and revealing the dynamics of evolutionary change. These advances continue to deepen our understanding of plant evolution, while highlighting the remarkable diversity and adaptability of plant life. The study of plant evolution and diversity provides fundamental insights into the history of life on Earth, while revealing the processes that have generated the remarkable variety of plant forms and functions we observe today. This knowledge is essential for understanding ecosystem function, predicting responses to environmental change, and developing strategies for conservation and sustainable use of plant resources. As we face unprecedented environmental challenges, understanding plant evolution becomes increasingly important for maintaining the plant diversity upon which all terrestrial life depends.